So you'll open RStudio, and RStudio is just an IDE, which stands for, I actually don't know what it stands for, but it's basically just a wrapper, a user-friendly wrapper for R. So anytime you're programming in RStudio, you are using the R programming language. So open RStudio, and then thank you, Natalie, integrated development environment. So from your downloads, open which, the RMD of your choice. So again, if you want sort of the blank one we'll fill out, you can open the workshop template. If you want the filled out code, you can open the workshop answers. And it will look like this. So if you're not familiar with an R Markdown file, that's what .rmd stands for. If you've only used .r scripts, I only found out about R Markdown scripts like 2 years into my tenure at Stanford, and it was life-changing. I almost never, if I'm writing in R, I almost exclusively use R Markdowns. And the reason, there's a few reasons, but one is because after you're done writing some code, you do what's called knitting, which effectively restarts the R session and runs all of the code in your report and generates a really nice HTML report with all of your code and all of the outputs. So it's a really easy way to kind of like, it's like a coding lab notebook, makes it very easy to share code and figures. I'm just all for it. So we're going to use an R Markdown file today. If this is new to you, it's not much of a jump from just an R script. It allows you to have text in between code chunks. So here you can see me using Markdown formatting to add headings and bold, etc., which will render when the Markdown is knitted. But these gray chunks are where the code happens. So for each code block, you have these 3 backticks and the letter R to indicate that it's an R code chunk. But before we even get to the workshop, I'd like to direct your attention over to the websites for these R packages, which we've put a tremendous amount of effort into documenting these R packages. So I just want to show you kind of what's available to you there. And the way to get to this is, unfortunately, I have not figured out Google's algorithm. And if you search for MoTrPAC rat training data, you will not find this package. But if you go to GitHub and search for MoTrPAC, you'll eventually find it. So here we have the MoTrPAC rat training data package and the analysis package. I have these bookmarked, so I'm going to first go to the data package, actually. And the reason we have two separate packages is because if you've installed these packages, you'll have noticed that it took quite a while for the data package to install, something like 10 minutes. And we don't want people to have to do that often, or really ever more than once. So the data package is just that. It has all of the underlying data, whereas the other lighter package has all of the functions that wraps that data and makes it easier to access those data. But you could actually use this package by itself to explore all the data you want. So this is what the GitHub repository looks like. This is where, if you're interested, you can actually look at all of the source code. It's all right here. And I would also stress that if you ever intend to use this data package, I highly, highly, highly recommend watching the repository. That means creating a free GitHub account, going to this drop-down menu, and for you'll have an option to watch. You can go to custom and select releases. This is because you'll get an email notification whenever there's a new release, which for the data package could mean changes to the underlying data. There's no other way for me to reach out to people who've downloaded this package and let them know if some major change happens or if we find a bug or a problem, etc. The only way for you to help yourself is to watch and click releases here so that you get an email if there's changes to this package. And again, if you ever plan to use this package, I would stress that. So each of these GitHub repositories also has a website, which you can find in this link here, and that's the sort of prettier version, and for most of you, the only version you'll care to look at. So you could bookmark this if you're interested. And there's a few tabs here. The home page is kind of has all the general info. You can see the table of contents here. There's a Get Started tab at the top that has a very detailed vignette that has a lot of detail about everything I'll talk about today. There's also this is, again, there's separate vignettes for the data package and the analysis package. So this vignette for the data package shows you how to use the data even without the other MoTrPAC package. There's also a reference tab, which is we'll be looking at quite a bit today. It lists everything that's included in the package. So in this MoTrPAC rat training 6-month data package, we can see all the kinds of data that are included. For example, metadata, we have phenotypic data, and we can click on any one of these links to get expanded documentation. So for example, let's say we're interested in the phenotypic data. I can click on pheno, and it tells me, okay, what this is. It's phenotypic data, blah, blah, blah. It gives me a description for every single column in this table, which for this guy, this is a lot. This is all manual, but it has everything here. So if you're interested in the specifics of any data object or function, this reference tab is for you. And just to browse a little bit and see what data we'll have available to us and what we'll be working with today, I'll just scroll through this page. So we also see that we have ohm-specific sample metadata. So for example, if you wanted to get some of the RNA-seq-QC metrics, you wanted to see the RINs, you could look at the transcript meta object. There's also there's a lot of sample level data, which there are easy ways to access through the other package. You don't actually have to know the names of these objects, but they exist and they're kind of what are in the back end of this analysis package we'll use. We also have, so this is just listing all the normalized sample level data. We also have all of the differential analysis results. And again, there are functions in the other package that easily retrieve a specific object of interest. You don't actually have to know each name of these objects, but it's all here. There's also a section of all the inputs and outputs for the graphical clustering I keep mentioning, as well as the pathway enrichment analysis. So I think maybe it was Tyler who asked the question about where we can find the underlying enrichment results for those networks of pathway enrichments we looked at. That would be the graph pathway enrich object here. We also have a bunch of feature mapping tables. So we have a feature to gene map that maps all the MoTrPAC feature IDs to genes. It's actually, I recommend using the smaller filtered one that excludes the epigenetic data because it's a lot literally smaller and easier for R to use. There's also the mapping of rat to human orthologs that we used in our analyses, as well as rat to human phosphocytes, metabolite feature annotations. So mapping those to KEG IDs, etc. And a bunch of other stuff. So if you're curious kind of what's in the data package, it's all here. The changelog tab also tells you what happens in each version change. So this is if you watched the GitHub repository, whenever there's a new version, you would basically get an email that had a note of what changed. So that, for example, in this version 1.7.0, we realized that this is where we updated the phenotypic data, because there was a consortium-wide email saying that the VO2 max data was from the sort of the data freeze. There were some issues. So this is the version where I updated the VO2 data. So this is an example of why it'd be important to watch the GitHub repository for changes. Now I'll move over to the analysis R package, which is kind of the front end, if you will. It's the package that has all kinds of functions to work with the data. So you don't actually have to work directly with, like you don't have to know the name of every data object you're working with. Similarly, this package also has a Get Started tab that has the basics, installing, getting help, etc. Now, in this case, the reference tab has information for every function available in this package, whereas for the data package, it's all data objects. Now we have the documentation for every function. And you can see on the right-hand side, the different groups of functions we have in this package. So first, to my previous point, there are a bunch of functions related to loading data. So for example, load sample data. This function, I can click to see more details. It says load raw counts or normalized sample level data. And there's all kinds of options to do different things. So we could, for some cases, like for gene expression data, we could say that we don't want the normalized data, we want the raw counts. Or we only want to include training regulated features in the output, not all features. Or we don't want to, we do want to exclude the outliers that we excluded in our differential analysis. So in this documentation, you can kind of really get into the weeds of what each function is capable of doing. And it gives you a description for each argument of what it means, as well as a description of the output and some examples of how to call that function. So these reference tabs are kind of the Bible for using these R packages. So I'm going to leave, just kind of leave this on the side here, since we may want to use it, and move over to RStudio. And see if I can, is this large enough for people to see both of these? Take that as a yes.
Yes, yes.
All right, so I'm working on, again, the template version of the Markdown, which has a lot of things empty, blank for us to fill in as we go. You may follow along in the answers template if you prefer to just run existing code. This first block is just loading a bunch of packages that we need to run analyses in this this Markdown. So in a Markdown file, you can run a single line with Command Enter like that, which is very convenient. Or you can run all of the code in this chunk, in this gray it off box, using the play button on the right-hand side. And you see this little bar shows you kind of its progress through the code, and is nice. So go ahead and load packages. And if you have failed to correctly install packages or haven't installed one yet, it'll give you an error and a little red bar and some message. Try to follow the error to resolve it. But hopefully these are all listed in the installation instructions, so hopefully no one has issues there. Then there's some text. I'm not going to go through this because we already have, but this will be rendered in the HTML report when we render it, and it's just kind of nice for reference. If you forget something, you can scroll up and see that it's there. It just tells us about these two R packages. And a note that all the data objects in the data package are in all caps, which is just kind of a nice way of indicating that, okay, I'm working with a data object. And we can also get the same reference that I'm looking at on the right-hand side here by using this question mark command thing in R. So here down in my console, this is where we run our code. Let's say, for example, I wanted to look at the documentation for that load sample data object. I start typing, and any object or function that's available in my environment from the packages I've loaded kind of already start populating. And once I see what I want, I can just hit tab to autocomplete and hit enter. And now on the right hand side in the help tab, it's pulled up the same function reference that we were just looking at on the web. So that's another way to get help for a specific object or function, is to use that question mark. Okay, then there's some text about MoTrPAC, which you all know, and study design, which we went over. Another thing you need to be aware of to use this package is the different tissue and assay abbreviations, because whenever you're kind of looking for results or asking the package to fetch a specific kind of data, it's going to expect you to use these abbreviations. So sorry if you have not used them for the last 2 years, you will have to learn them a little bit, but they're all listed here. And you can also list them in these objects in the data package, list them all as well. So you can just type and enter tissue abbrev and it tells you all the tissue abbreviations, or assay abbrev and it tells you all the assay abbreviations. And a nice feature of our Markdown is that it has all the output for your code right under the code chunk instead of in your console, which is just really lovely. Okay, so with that, the guiding question for this tutorial will be to see if we can use these R packages to identify an interesting shared multiomic signal in the heart and gastrocnemius muscle tissues. I'm going to use interesting shared signal rather loosely. I'm really not a biologist, but I'll leave the biology to you. So let's start with some sample level data and looking at how we can get sample level data. I'm going to expand this a little bit because I'll actually use my help viewer to get some of the documentation. So we're going to use this function load sample data to load sample level data for a specific ohm and tissue. So again, I can show you, use question mark in the name of a function. It pulls up the documentation on the right-hand side here. So let's say we want heart protein expression data. I'm going to call it heart prot. Minor aside, it's in R, it's sort of the standard to use what is it, less than dash as like an equal sign, but functionally it's not different. This is just my, I do as I say, not as I do, but I'm going to use equal sign because it's less typing, and I don't like why I don't like that R does that. But so I'm going to use equal sign. You can use your less than dash. But you can see that when I start typing load sample data, I get this little help thing here that also tells me what the inputs are for this function. So I said I wanted the heart protein expression data. So I'm going to say heart, which is the abbreviation for the tissue. And I'm also going weird, Tyler, thanks. That's interesting. And then the assay abbreviation I want is prot. And then I can look at the rest of these default arguments. I do want the normalized data. I don't want only the training regulated features. I don't want to exclude outliers for now. And that's all we need. So, okay. Now I'm gonna, again, it's command, or on my keyboard, it's super, or see Windows, it's the Windows button. But to run a single line of code, it's super command Windows and enter. And then I'm just gonna kind of show you so that it's easy to apply to any combination of.
Hey, Nicole, I have a question.
Yes.
Why, in addition to your object, it created a second object with a different name as like automatic output?
It did. Yeah. So I don't know why this happens. It's the way I'm fetching the data from the R data package. When you basically, so there's something called lazy loading, which when you load your you run this command, you say library MoTrPAC rat training 6-month data. It kind of tells R the list of all objects in this package without actually loading the data itself, because if it loaded everything, it would kind of overwhelm, it would be too much memory. But it's kind of it's ready to be grabbed, basically. So if I do, let's say for example, I start typing feature to gene, R knows that there are these data objects from the data package available with these names, even though they haven't actually been loaded into memory yet. That's what lazy loading is. But then when I actually call an object, it is, oops, I didn't call it, I just asked for the docs. If I call an object, oops, why not? I expected it to come in my environment, but weird. Well, I guess the answer is I don't really know.
I think that this is because
with...
you are running it on the Mark and the RMD. If you run it on the console, it's gonna run it there. Yeah, you should see.
Weird. Well, it's inconsistent then.
Yes,
I know
just only the object. Yeah.
If I actually call data, then it will load it into values, and you see this promise evaluation. It's also because it hasn't actually like this. I feel like this is a bug in R, because I've worked with this and some of these objects are loaded and then R starts getting full. So I clear the workspace and then I no longer see any data objects, but there's still like 2 gig being used in memory. So it's a little bit of an oddity. Short answer is that this is actually showing you the name of the object in the data package that is being fetched and reformatted if necessary to return you what you asked for. And I have tried to prevent that from happening and I can't figure it out. So that's what that is. But you can ignore it or set it to null to remove it from your environment. Actually, let's see what happens. So right now there's 570 megabytes being used by my environment. If I say actually, I don't want to set it to null because then it'll change environment variable. So anyway, good question. Short answer, I don't know how to not have that happen, but it's an artifact of having an object sort of loaded from the data package.
I ran into that issue the other day, so I can send you the code so it doesn't load the actual object that it's trying to pull.
Oh, awesome. Who is that speaking?
Tyler.
Tyler, thanks. That'd be great. Yep. Okay, so I'm just gonna copy and paste from the answers to make sure that I have the right objects being called here. But this is how we load sample level data for any specific tissue and ohm. So let's look at and also when you call load sample data, it prints a message with the name of the corresponding data object that it's grabbing from the data package, just kind of for your information. If you want to know, like this object has all the normalized heart protein data, whereas all the normalized immunoassay data is from a single object called immuno norm data flat, and same for metabolomics, all of the normalized metabolomics data are in an object called metab norm data flat. So just giving you some information. Now let's look at the format of these tables, because we've loaded the data, but we don't know what we're looking at yet. Oops. Gonna look at our heart protein data, and you'll see that this first column called feature is missing, or NA, not assigned, for a number of features. And that is deliberate. There's some text below here that says the feature column is non-NA only for training regulated features, which provides a convenient way of identifying this subset of features. So for all of the sample level data and differential analysis results, if there's a feature column, that value is only defined for training regulated features. And again, this is part of the reason for this is because we needed unique feature names to do the graphical clustering algorithm, which is done only on the differential features. So it actually kind of causes some problems if you try to define the feature for everything, but also is a handy way of quickly identifying just training regulated features. Then we have a feature ID column, which is just the MoTrPAC feature ID. Tells you which tissue we're looking at, which assay, and then all the remaining columns are the sample level data. And in this case, our column names are vial labels. If we look at, for example, the gas shock metab data, we notice something a little different. The column names are now actually PIDs. These are participant IDs. So it's one feature per, sorry, one ID per animal instead of one ID per sample. And this is because of the redundant nature of metabolomics, where we have multiple measurements per some features, and it was easier to put all the data across all platforms into a single table. So just be aware, sometimes because of assay specific issues, sometimes the column name is a PID, sometimes it's a violabel. Sometimes it's also, so, you know, it's nice to have the information in these tables. For metabolomics, you can also see which data set or which platform it's from. But oftentimes we just want strictly numeric data frames, and it's kind of annoying to have to deal with these other character columns when we want to do, for example, principal component analysis. So there's a function called df or data frame to numeric that does this for you, even though it would only take a couple lines of code. It's just kind of convenient. So let me just show you what that does. I'm going to call df to numeric on my heart prot data frame. And you can see that there's this row names argument that's basically asking which column in the existing data frame do you want to become the row names. So actually, I'm going to use that default. I want feature ID to be the row names. And we can see that it returns what we expect, a numeric matrix where the feature ID are now row names. So that's just an easy way to convert it to sometimes a more conducive format. Okay, so we already talked about this. So now let's plot the sample level data for a single differential feature. We're going to use the plot feature normalized data function. We can look for the documentation for this function using that question mark. So this function is to plot normalized sample data for a single feature. Okay, so let's choose a differential feature in the heart. So let's again, we look at heart, our heart proteomics sample level data. Remember that feature is non-NA when the feature is differential. And let's say we want to look at it as a differential feature. So let's just look at this specific feature. So we can copy and paste it. And we're going to say plot feature normalized data and input feature as this string. Alternatively, we could use the assay, tissue, and feature ID arguments to say assay equals prot, tissue equals heart, feature ID equals this guy. But we'll just use feature. And there are some other options here. We can add the gene symbol, which I would like to do. So I'm going to say add gene symbol is true. And let's run that line and see what happens. And as long as it's green, it's sort of thinking it's running. And this is our result. So we see nicely in line, we get this is the normalized sample level data for this particular feature, which we know is differential because of how it's shown in the table. There's actually also an option to, I think, add adjusted p-value from the differential analysis. Let's say I also want to add that p-value. By the way, this is a very new feature. So if you installed the analysis R package more than like a week ago, you might have to reinstall it to get this newest version. But if I add that argument, add adjusted p-value, we also now see that, the adjusted p-value is added. I guess this isn't too helpful. Maybe I should do 2 significant digits, but this means that it's less than 0.000, I think. But I'll add that as a note to add significant digits. Okay, any questions so far? Okay. So, okay, we've explored the sample level data a little bit. We can also use, there's another way to retrieve sample level data, which is this function combine normalized data, which we can use if we want to load data from multiple tissues or multiple assays. So in the examples above, we wanted specific data sets. So we used the load sample data function for specific tissue and ohm, but we can use combine normalized data to download multiple data sets simultaneously. So an example here is that we can use this combine normalized data function, specify gastric nemesis as our tissue, and importantly, if you're going to use this function, setting, I think this is, let's see, this is already, I believe it's the default. I just wanted to remember to mention it. Let's see. Include epigen as by default is false, but just to be clear what that's saying, the epigenetic data sets are very large. There's like hundreds of thousands or millions of features per tissue for ATAC-seq and RRBS. So if you unknowingly requested to download epigenetic data for a large number of tissues, you could easily overwhelm your RStudio session. Will probably crash. So you can download epigenetic data using the combine normalized data or load sample data functions, but you have to specifically specify to include the epigenetic data, which in the back end is actually downloading the data from Google Cloud Storage to your local computer, reading it in from your local computer, deleting it from your local computer, and returning it to you. The reason for that is that if we actually included the full epigenetic data sets in the R data package, it would be monstrous and impossible to use. Any questions about that? Okay, so I'm going to run this chunk that's already filled in just to kind of show what's happening. So first, it tells you that it's going to exclude the epigenetic data, which we're okay with. And then it lists every other data type it is collecting. And I asked it to tell me how many features there are in total. So there's 50,000, over 50,000 features. We have the sample level data for gastroc now. How many are differential? We can quickly ask by looking at how many rows does feature exist. There's almost 2,300 features that are training differential in gastroc. And what ohms do these differential features belong to? We can just look at a table of the assay for those rows where feature is defined. And we see that most of these differential features are at the level of protein phosphosites and proteins, as well as transcripts and some metabolites. Any questions so far? Okay, I'm going to try to, I will try to end this by 2, since that was the scheduled time I had in your calendars. So I'm going to skip some things. I'm not sure what yet, but I do want to get to the meaty stuff that I know some of you are interested in. So we're also going to just quickly look at the head of this combined data, and we see again, column names have been changed from vial labels to PIDs, because now we have multiple measurements across in the same animal across many samples now. So instead of having one column per vial label where each of those columns would be mostly missing, we combine data from the same animal, same tissue into the same column from multiple ohms. I don't think principal component analysis is the most interesting thing, and that's not something that's unique to this package. So I'm going to skip this section. We can come back to it if there's time, but this is just to show, how you can easily format the data to be readily input to PCA, and the answers doc has the full code for how to neatly and prettily plot those results using the color codes from the data package. But I'm gonna ignore this for now, and I just set this chunk setting eval to false. And what that means is that when we knit this document at the end of this tutorial to make the HTML output, this means don't evaluate this chunk, don't run this code, because if I did try to run this code, it would throw an error and the knitting would fail. So it's just going to ignore this chunk of code when we knit to HTML. And this one too, I don't want to perform PCA, done enough of that. So set eval to false. But again, if you'd like to see how to do PCA and plot the results with our tissue and sex colors, etc., you can look at the answers. Okay, so now we've looked at the sample level data. Now let's look at differential analysis results. And just a reminder, there's the 2 times the time-wise and training differential analysis results. In general, people pretty much only interested in the final adjusted p-value from the training differential analysis, which is already included in which we merged with the time-wise differential analysis results, so that everything you most likely want is available in sort of the default way I'll show you. If for some reason you are interested in the other training differential analysis summary statistics, which include, for example, the male and female specific p-values before we merge p-values per feature, you can load those summary statistics again from Google Cloud with the load training DA function. But I say that as kind of an aside because I doubt any of you will want to do that. So we're just going to use the sort of main results that are available in the package. First, looking at First, looking at this training regulated features object. So this is it's in all caps, which is an indication that this is a data object in our data package. So let's just look at what this looks like. We see the feature column is populated because these are all significantly regulated, significantly differential features now. So we have the feature column, the assay, as well as the assay code, which is still used in the BIC release, the tissue abbreviation, as well as the tissue code, which is also still used in the data release. This non-redundant feature ID, I won't get into now, but it's related to repeated features from meta-regression. For metabolomics features or immunoassay features, it tells you what platform the data are from, as well as the specific sex and time point the result is from, and then all the summary statistics. So the time-wise log fold change, time-wise p-value, time-wise z-score. These are the inputs used for the graphical clustering analysis. And there's a whole bunch of other stuff too. And then the training Q, that is the adjusted p-value from the training differential analysis that we use to select training regulated features. So reminder again that we only have one of these adjusted p-values per feature from the training differential analysis, which is why it's just the reason we see the same value for the top 6 rows here, because these are all results for the single feature just at these different sex and time points. So all eight rows in this table for a single feature, there's 8 rows because we have 4 time points and 2 sexes. All 8 rows will have the same training Q value, and that's the value we use to select training regulated features. So for every row in this table, they'll all have a training Q of less than 0.05, because that's the threshold we set to select training regulated features. So any questions about this? Because this is probably one of the most useful data objects. Okay, so if we're also interested in the full set of differential analysis results, not just the significantly differential one, we can use this function called combine DA results. For example, for single tissue, this would concatenate all the differential analysis results from heart. This takes up a bit of memory, and I'm already at 2 gigs somehow, mostly because of this feature to gene guy. I'm gonna just try to remove memory consumed by a feature to gene, but to my point, I think it was Natalie who was speaking up earlier. I don't think it's going to reduce memory consumed anyway, which is just a bug of R or RStudio or something.
I think you have to run GC as a follow-up, just because I don't know why it doesn't
That might
do.
not even work. Oh, that time it did. Okay, great. I've messed around with this. Sometimes there have been times that my environment is completely empty. It's using like 2 gigs of memory and running GC does nothing. So I have not figured that out yet, but thanks for that for now. That works. So I'm not going to actually run this this chunk because it takes quite a bit of memory, and that's why I also have this chunk set to val equals false. But now let's plot the differential analysis results for a single training regulated feature. I was going to subset this up here. Okay, so going back for a chunk, I'm just looking at my own answers. I wanted to show an example of looking at just differential features in the heart and gastroc, because that's what we were interested in looking at today. So we can easily subset this table by saying, for example, the tissue column is either, so it's in heart or gastroc, all columns. Okay. Also, another sort of aside, most people use either tidyverse or data table. I'm using neither. I'm using base data frame just for the purposes of today's tutorial. I wanted to use base R, but in practice, I always use data.table. To me, it's much more intuitive. But in general, people will either use tidyverse is also very popular. And there's, you would do this subsetting a different way with either data table or tidyverse. But again, today, I just wanted to use base R for the sake of broad audience. So we can ask easily how many differential features we have in this tissue. 47,000, that's a lot between the two. And we can ask, for example, what assays and tissues those belong to. So we can make a table of tissue by assay. And we see a lot of differential features in level of protein phosphocytes, proteins, transcripts, metabolites. Oh, I know why I did. This is not the number of features. This is, remember there's eight measurements per feature here. So that's why I thought that was an awfully large number. So this would actually be, let's see, we could do something like, well, we could just
search for the unique values, right?
Yeah. Well, if we took unique rows now, it'd be the same, but we could subset the columns and then take unique rows. But just for now, purposes of showing, we can subset these results. So now going down to plotting differential analysis results for a single feature. Let's again look at our subset of differential features in the heart and gastroc. And let's just grab the first feature we see. And we'll use this plot feature log fold change function. And again, use the feature argument to pick a feature. And this is protein acetylation. So I do want to add a gene symbol. And what else? I'll show you a couple other things we can do, but for now, let's just plot this log full change for this features. Okay, great. We can see males and females, the adjusted p-value. I think it looks nicer to facet by sex. So we can say facet by sex is true. In which case now we see females on one side, males on another side, and now the lines are colored by tissue instead of by sex. I don't know, sometimes we want to do this for, if there's space constraints, we can also say scale x by time is false. So right now, the x-axis is scaled by time. But if we say scale x by time is false, then they are just equally separated and makes it easier to make the figure smaller if we need to squish it into a small space. But that's how we can look at a single feature. And now, okay, so we've looked at sample level data, we've looked at some differential analysis results. Just again, briefly want to mention that combined DA results function is how you would get all differential analysis results for a tissue and or ohm. Even though we just looked at significant features here. So now let's move to the graphical clustering results. So this is getting to the kinds of figures I was showing you in the Jupyter Hub. And we're already at 153, so definitely not going to get as far as I thought today, but that's okay. So I'm going to try to focus on just the most interesting things. So first, let me show you how you can make one of those, the plot of the actual graph that I showed in the first tab of the Jupyter Hub notebook. And in this case, because we're time constrained, I'm just going to copy and paste the code from the answers. And just for a single tissue for now. And you can see these chunk options at the top change how large this figure looks. So like right now, this is the text is too small. It looks weird. This has to do with this sort of, it's system specific. It has to do with the resolution of your screen and also how zoomed in you are and all weird stuff. But you can mess around with how large this figure is to you. So here, again, don't have time to go into each of the arguments today, but you can look at the documentation for this get tree plot for tissue function to see how to easily make a tree plot of your own with any number of trajectories, any combination of tissues and ohms. You can show parallel edges by ohm or parallel edges by tissue, change the curvature, edge alpha, all the things we did in the dashboard. Now you can do with this function as well. The dashboard is actually a wrapper for this function. Questions about that? I just generally, yeah, I guess I didn't leave not much to ask questions about, but that's the function you can use to plot the tree plot. We can also use that same function to plot features for both of these tissues together. So I'll just show quickly what that looks like. And this is setting parallel edges by ohm to true. So you'll see what that looks like. But again, we can list multiple tissues and multiple ohms to be included in this plot. And here we see that for these top three trajectories, the ohms corresponding to each one. So you can play around with this and plot whatever combination of features you're interested in. And I note there's a lot going on in each of these three trajectories. We'll focus on these top three largest paths in the heart and gastroc for the rest of this workshop, which I will go a bit after too, if anyone's still on, but don't expect anyone to stay on if, 2 hours is a long time. So there are ways to get features underlying the graphical clusters. I'm going to just show you the main, most useful one probably is using this data object called graph states. So just briefly to show you here, we have one row per differential feature now. It tells you, and if we scroll over, it tells you the graph states for that feature at each time point. So this is why it was important to understand what those graph states meant, because all this information is condensed and easily summarized in this graph states object. So for example, this first feature is null through weeks 4 in both sexes, and then between 4 and 8 weeks, it becomes downregulated in both males and females. And here I show an example of how we can extract the features for any given path. So for example, here I wanted those 3 specific paths we were looking at in this cluster. So I copied and pasted the names of the paths. And this is a bit of code to how to get a named list with all the features in those paths. So using this graph states table is probably the easiest way to kind of figure out what features are underlying any given graphical cluster of interest. These are functions that you can use to, again, reproduce plots that I showed you in the Jupyter Hub dashboard. So I will skip these just to say you can use these functions to do that yourself. So this plot features per cluster will show you the bar plots of, here, let's see if I can quickly do that. I'm gonna run this code to get some sample data to work with. And I'm going to call that my clusters. And then we can call plot features per cluster on this object. The sizing is really weird again, so let's see if that's better. Okay, so now we see that for these trajectories in gastroc and heart, so the reason it's split by tissue is because I actually split the paths by tissue. So I'm missing, this was not the case in I think I just copied and pasted the wrong code. Yeah. But anyway, I'm gonna move. Yeah, feel free to jump off. I just want to show, I think, the last bits of things about pathway enrichments, running your own and plotting the networks, because I think that's one of the most powerful things that the package can do in terms of helping you explore results, but feel free to drop off. So as I mentioned before, this graph pathway enrichment object has all the pathway enrichment results. So we can take a look at it. It has a lot of columns. So here is the list of all the column names versus the data table. And you can also use the documentation to get definitions for all of those column names if you're not clear what something is. But this is all of the pathway enrichment results. So I'm just going to subset. Let's see if anyone stays on first, but thanks for everyone who attended. So I just briefly show an example of how we can subset these results. Let me just copy and paste from the code to make sure I have the right clusters object here. I think I do. So I'll show an example of, here we're taking the pathway enrichment results, we're subsetting it to using the cluster column to just the clusters I'm interested in, which you can see here on the row names. And also selecting significant results at 10% FDR using the adjusted p-value column. So we can see all the significant, the number of significantly enriched pathways for each tissue and cluster here. So we see, for example, a large number of significant pathway enrichments for features upregulated in all 4 time points in the heart. If you want to perform your own pathway enrichment, there are several functions to do that. The main one is cluster pathway enrichment and is intended for performing pathway enrichment on on multiple groups of features. You can again, look at the answers to see how to do that, and read through this text. Gives a detailed explanation of what it's doing, what your options are, and what some of the messages mean. And looking at significant results. You can also perform enrichment with a single list of genes using the gene pathway enrichment function. So this is just for a very simple analysis. If you had a specific set of specifically genes you're interested in performing pathway enrichment for, it's a simple wrapper for gprofiler ghost function, which is what we use to perform all the pathway enrichment for the manuscript. So here's an example where we're selecting features. We're selecting differential features in differential proteins in the gastroc that are upregulated in both sexes at all time points. That's what that line is doing. And then we're using the feature to gene map to map those feature IDs to gene symbols. So if we look at features, they're kind of these are, I think, uniprot IDs or something, which gprofiler doesn't do well with. So then we map to genes, and we can look at input, and we see now gene names. And then we'll specify our background using this other object in the data package that is described in the text above here, but it specifies all of the proteins that were tested for differential analysis in the gastroc, which is an appropriate way to specify the background. And a word of sort of caution: if you're performing any of your own pathway enrichment analysis, it's important to specify a background and not just let it be the default, because then you'll get artificially small p-values. Think of it basically, if you're asking to enrich a list of genes that you found were, significantly regulated by training in muscle, and you didn't specify a background, your tool is going to assume that all annotated genes are the background. It's just something like, in rats, it's something like 30,000 genes, when in actuality, there's only about 15,000 genes expressed in that tissue. So by not specifying the background, you are saying that your result is kind of, if there is some overlap with a pathway, you're going to get a more significant p-value than is really accurate. I didn't do a good job of explaining that because I'm trying to rush, but there's some details in that in the text as well. And we can use this gene pathway enrichment function to just quickly run pathway enrichment for this small set of genes, and then look at the outputs, and we see all these significant pathway enrichments for that gene set. So cool, easy wrapper if you'd like to do your own pathway enrichment analysis. There's also a function if you want to use custom pathways, and I provide an example here of using MoTrPACs, but I'll skip this for now. And the one more thing I would like to share is how to make these interactive networks of pathway enrichments, which is the last thing I showed you in the Jupyter Hub portal. And importantly, is something that you could do with your own pathway enrichment results. Does the gene enrichment wrapper work for Fella? No, but the cluster pathway enrichment function does. So you could actually just use the cluster pathway enrichment function with a single group of genes, and it would work fine. And there's some extra steps you have to take if you want to do enrichment for metabolites, which are included in the documentation. But yes, Dan.
So quickly, one more question, Nicole. What is the difference between cluster pathway enrichment and gene pathway enrichment? Maybe if you have description, I can read it later and get back to you. I don't want to consume more time here.
Yeah, if you could, so the short answer is gene pathway enrichment accepts a single list of gene IDs, whereas cluster pathway enrichment accepts a table of MoTrPAC feature IDs that specifies which tissues and ohms and graphical clusters those features belong to.
Okay, thank you.
Mm-hmm. Okay, so this is the last part or sort of step of the tutorial I'll share. And I just do want to take a moment to share this because it makes a lot of data accessible to you, and you can also run this enrichment network viz function on your own pathway enrichment results for any project, not just MoTrPAC. So I do want to share this. I'm gonna see, look at my answers to make sure I'm doing this. So basically the input for enrichment network viz is a set of enrichment pathway enrichment results. So I'm gonna just copy and paste some code in here so you guys can see what is happening. But basically, again, if I'm reminded of the clusters that I was interested in looking at, I can copy and paste one of these cluster names to select or subset the graph pathway enrichment results to those corresponding to this specific cluster. So that's what I'm doing in this line. I'm just taking a subset of pathway enrichment results from the graph pathway enrich object. So this is, something like this. There's more details in the documentation, or I guess something, a table like this is what the enrichment network viz function expects. And you can see it's effectively the G profiler output with an additional cluster column. So again, you could easily reformat your own pathway enrichment results to be acceptable input for this enrichment network viz function. So I will input those pathway enrichment results. I'll specify the title that I want to be shown in the plot. Run this. It might take a few seconds. But what it's doing now is like computing similarity between all these different pathways to construct this graph. And it took 15 seconds, which is longer than most things. But now we have the same kind of interactive plot that I was showing you on Jupyter Hub, except that you can do it for any subset of pathway enrichment results that you're interested in looking at. So again, we have colored clusters that indicate groups of pathways that reflect similar biology. Like here we have carbohydrate metabolism. In bright green, it's just metabolism. Teal or whatever, neurodegenerative disease, and it's just as interactive as the Jupyter Hub. We see the pathway name, its parent, p-value, the data sets for which the pathway is significantly enriched, and the corresponding genes driving that enrichment. So that's the last thing I wanted to show you today. And I think, I'm my very biased opinion, I think is a cool tool that you could use for your own research as well. So thank you all for joining me today. Are there any last questions? Or before that, I'd just like to say again, you can look at the workshop answers are marked down to look at this completed code and play around with it, do whatever you want. And I hope you found this useful. Are there any other questions? Okay, great. Well, if you have any suggestions for, additional analyses that you think would be useful, I'm giving a tutorial for this R package at University of Florida in a couple of weeks. Obviously, I ran out of time in the second half, so I need to trim it down. If you have suggestions for things that specifically for the R package part that you think would be really valuable that I didn't do, or things that you thought were less useful, please let me know. I'd really appreciate that. I'm also going to send a survey that I'd very much appreciate if you filled out so I could help improve my workshop that I'll give to Karen's group in a few weeks. And I will let you know where the recording is available as well. So thank you all for joining. I really appreciate you sticking with me for over 2 hours.
Thank you, Nicole.
You're welcome.
Thank you, Nicole.
Yep. Thanks so much, Nicole. That was incredible.
I'm glad it was useful. Thanks, Jim.
See you later.
See ya.